Welcome everybody. It's my great pleasure to welcome you all for the second lecture in our short series, the Live Theory Lab 2020. It's my great pleasure that Charles Nelson agreed to give the presentation today. As in the last week, I would like to ask you to use the GoToWebinar chat function, probably on your right somewhere, to pose questions throughout the talk. My co-moderator, Miriam Sander, will edit and curate the questions throughout the talk. After the presentation, we will have a short question and answer period. So you can also use this raise your hands option to indicate if you have a question to our speaker. Again, Miriam and I will curate the question and will offer you the possibility to post your questions via audio. Please keep in mind again that the whole session will be recorded, so if you and potentially will be made available on our YouTube channel. So if you prefer to stay anonymous and would not like to appear on the recording, please keep your mic muted throughout the whole session. Again, as a good tradition in life, I would like to ask one of our fellows, Mareike Trauernicht, to introduce today's speaker, Charles Nelson. So please, Mareike, this is now all yours.
Thank you, and I'm honored to do so. So for the introduction of our speaker today, I could basically fill the whole session because the CV is more than 100 pages long. So I really tried to filter the information that are most useful for us maybe today. So Charles Nelson received his PhD in child and developmental psychology from the University of Kansas and did a postdoctoral training at the University of Minnesota. He took his first faculty position at Purdue University in 1984, then at the University of Minnesota in 1986. And in 2005, Professor Nelson moved to Harvard University and Boston Children's Hospital, and now he is a professor of pediatrics and neuroscience, of psychology and psychiatry, and of education. Further, he leads a research network on early experience and brain development, and he was elected into the National Academy of Medicine in 2018. His research interest is in the relation between brain and behavioral development, and with the use of a variety of neuroimaging tools. He focuses mainly on three topics. First, on the development and neural basis of processing social information, for example, face recognition. Second, on trajectories of autism. And third, on effects of early adversity on brain and behavioral development. We are very pleased to have you here with us today. Thank you very much, and we are very much looking forward to your talk. So the virtual stage is yours now.
Okay, thank you very much for that gracious introduction. So let me begin with an outline of what material I'll be covering today in my talk. So I'm going to begin with maybe spending just 2 or 3 minutes of an overview of prenatal brain development, because that will set up the rest of my talk, which is really focused on plasticity and critical periods. Then I'll turn my attention to how experience impacts brain development. I'll define what we mean by a sensitive period versus a critical period. I'll talk about neuroplasticity. I'll very briefly talk about the biology of sensitive periods. I'm not sure how many of you are molecular neurobiologists, so I thought I'd keep that part brief. And then I'm going to give examples of critical periods and plasticity drawing from different domains, including visual development, auditory development, speech and language development. And of course, you're welcome to ask questions on other domains. I've spent many years studying the effects of psychosocial deprivation, so I can also talk about cognitive and social emotional development. And I'll conclude by talking a little bit about face processing, and again, in the context of critical periods. So let me start this movie. This is a brief movie that shows an animated view of what happens shortly after conception and then brain development through the prenatal period.
This animation shows brain development from conception through birth. In the first 4 weeks after conception, neural tissue known as the neural plate forms in the outermost layer of embryonic cells. The neural plate folds to form the neural groove, then curls to form the neural tube. This tube differentiates into the forebrain, the midbrain, the hindbrain, and the spinal cord. The forebrain will develop into the cerebral cortex, which translates sensory stimulation. It also controls complex behaviors, thoughts, memories, and problem solving. The midbrain will develop into a neural relay station for sending information from the body to various sites in the brain. The hindbrain will control the most basic physiological processes, such as breathing and heart rate. The spinal cord is the pathway for conveying information between the brain and the rest of the body. Between weeks 4 and 8, the embryo grows rapidly and the face becomes recognizably human. The eyes on the side of the head begin to migrate forward, and 2 distinct hemispheres of the cerebral cortex emerge. Between weeks 8 and 26 of the fetal stage, the cerebral cortex grows to cover the midbrain. By week 28, a major structural change begins. The cortex expands greatly in surface area and becomes wrinkled and folded inside of the skull. Beginning at week 28 and continuing through week 40, the brain surface fills with hills and valleys, called gyri and sulci. These cerebral folds and wrinkles help to create a vast neural network, which allows the brain to become a highly flexible yet precise control center and processor of information. By week 8, the structures of the brain are already in place, but the amount of brain tissue is rapidly increasing as many new nerve cells are formed. The formation of new cells is known as neurogenesis, or neural migration. During neurogenesis, new nerve cells migrate from the innermost layers of tissue outward. Like gymnasts climbing ropes, the young cells are guided to their final destination by fibers from another type of cell, the glial cell. Glial cells form the underlying structure of the nervous system. Although earlier formed cells rest closer to where they originated, later formed cells move further away from their starting point. In this way, the brain grows, adding layers to itself like the skins of an onion. Once each nerve cell reaches its destination, it begins to acquire characteristics specific to that area of the brain. At this point, each nerve cell sprouts the extensions it needs to communicate with other neurons. There are 2 types of extensions, dendrites and axons. Dendrites look like a network of antennas and receive incoming signals. An axon is a single, larger extension which carries outgoing signals to other neurons. Synaptogenesis is the process in which synapses proliferate in the brain. A synapse is a tiny gap between the dendrites of one neuron and the axon terminal of the next. It's the point of communication between 2 neurons. So many new axons and dendrites form during the process that it's sometimes called exuberant synaptogenesis. This cell growth process continues into a newborn's first year of life. For an information signal or impulse to move from the axon of one neuron to the dendrites of another, it must cross the synaptic gap. In this animation, an impulse looks like a green ball of light. When an impulse reaches the end of an axon, a chemical called a neurotransmitter is released. In this animation, neurotransmitters look like red balls of light. Neurotransmitters allow an impulse to cross from the axon of one neuron, across the synapse, and onto a receiving dendrite on another neuron. To create a more efficient and rapidly working brain, some of the newly formed synapses are eventually removed. Synaptic pruning occurs during the fetal period, infancy, childhood, and adolescence, It's believed that pruning is a way in which the brain becomes customized to an individual's nervous system as a response to that individual's unique experiences. This animation.
So I think by now that for those of you who don't have a background in developmental neurobiology, that was an entire course in neurobiology. And here's a brief summary of this, a slide I made many years ago. And this is demonstrating the very early formation of the neural tube, cell migration. And then this represents the peak of synaptogenesis in different regions of the brain, auditory and visual cortex, receptive language areas, and higher cognitive areas. And the fall off represents after the conclusion of synaptogenesis when adult numbers of synapses have finally been obtained. So now let me turn my attention to plasticity in critical periods. The term plasticity has generally been used two ways in neuropsychology. One is the recovery of function, whereby some function is returned following an injury, and a related concept is sparing, or the lack of loss in performance following brain damage. And both are often categorized as forms of functional plasticity. Neuronal plasticity is hypothesized to underlie functional plasticity. So when we think of these plastic changes behaviorally, the underlying neurobiology of that mechanistically is thought of as neuronal plasticity. So there are different examples of neuronal plasticity. Anatomic would be, for example, the ability of existing synapses to modify their activity by sprouting new axons or by an expansion of the dendritic surface, like new dendrites, new dendritic spines. Neurochemical might be the ability of existing synapses to modify their activity by, for example, increasing neurotransmitter synthesis and release. Metabolic might be fluctuations in cortical and subcortical metabolic activity, like glucose utilization, increased blood flow via increased capillary growth. And these are all examples of neuronal plasticity. So briefly, functional plasticity reflects the malleability of behavior to be protected following damage or to recover following damage, whereas neuroplasticity reflects the mechanisms that underpins functional plasticity. So an example that might be close to many of you who are trained in psychology is the efficacy of cognitive behavior therapy for the treatment of depression or anxiety is based on changes in thought pattern, functional plasticity, and these changes have to be mediated by changes in the brain, like building new neural networks. So there are four ways that environmental input and genetic constraints interact during development. The first is maturation. So these are behaviors that would occur without any necessary experiential influence, like a reflex. These are experience independent, and I'll give an example in just a moment. Maintenance refers to a fully established behavior whose continued expression depends on adequate and appropriate experience. So human infants produce and comprehend a wide range of phonemes in early babbling, but only exposure to a restricted set of phonemes will maintain those phonemes in the language. And I'll spend some more time talking about this in a few minutes. Facilitation is a behavior that can fully develop without appropriate environmental input, but which matures more rapidly with appropriate experience. So Esther Thalenlin and others have demonstrated that the onset of walking may appear sooner among infants receiving practice in walking, but the timing is important. So she's shown that if you put an infant on a treadmill just as they're showing signs of walking, you can actually facilitate the rapidity with which they learn to walk. And then induction would be the behavioral components will never fully emerge without appropriate experience. So children will not learn the elements of a native language without hearing the native language spoken. So moving back to a point made in the video I showed you, individuality is the product of both personal experience and biological inheritance. Genetics specifies the properties of neurons and neural connections to different degrees, in different pathways, and at different levels of processing. But because many aspects of an individual's world are not predictable, the circuitry of the brain relies on experience to customize the connections to serve the needs of the individual. Experience shapes these neural connections and interactions, but always within the constraints imposed by genetics. Experience is the product of an ongoing reciprocal interaction between the environment and the brain. So any given experience can vary enormously under identical environmental conditions, depending on the history, maturation, and state of the individual's brain. The relative maturity of the brain has an enormous impact on experience. So different areas of the nervous system mature at different rates, and lower level processing areas mature earlier than those at a higher level. A less mature brain is affected largely by more fundamental features of the environment, such as light or the speech train. And here we might keep in mind, for example, a baby born 2 or 3 months prematurely is now confronting the same environment that a full-term baby would, but the maturity of their brain is at a very different state, so they'll integrate that experience quite differently. So as the brain matures and changes with experience, more detailed aspects of the environment influence it. Thus, as an individual's brain changes, particularly during these early developmental periods, the same physical environment can result in very different experiences. So this leads us to our discussion of sensitive and critical periods and what we mean by this. So we think of a sensitive period as defined as a time in development during which the brain is particularly responsive to experiences in the forms of pattern of activity. So the point I'm making here is that although genes specify the properties of neurons to different degrees and at different levels of the nervous system, after birth in particular, much of brain development is sculpted by experience. And the periods of time when the brain is particularly receptive to experience is generally how we think of as a sensitive period. We might think of this as a critical period if the presence or absence of an experience results in irreversible change. And something that I've discussed for years is, in the human, can we think of examples of critical periods that are different from sensitive periods where the change is irreversible? And I think in the domain of behavioral development, we're probably safer to refer to these things as sensitive periods and not critical periods. So here's a slide I'd like to borrow from my colleague Tikau Hench that illustrates what we mean here. So we have in utero genes lead to the construction of the brain, and then we have environments that impact development in sensory or motor language or higher cognitive systems during these critical or sensitive periods. And then the result is the adult manifestation eventually of behavior. There are two important features of critical periods we should understand. Length is proportionate to lifespan. So the shorter the lifespan, the more rapidly a critical period comes and goes. And the critical periods occur in a hierarchical succession. And as you'll hear me say a few more times over the next little while, there is no one critical period. There are multiple critical periods, and there are cascading critical periods. And so, as you see in this slide, critical period for sensory development, like vision or hearing, occurs before that of language, which occurs before of higher cognition. And if you go back to the slide I presented much earlier, I showed you that these different systems remain open for different periods of time. So adolescent brain development is primarily a hallmark of changes in higher cognitive functions, whereas sensation and language have developed much earlier in life. The last point I want to make before I dive into my examples is that the ability to change the brain decreases over time. And here I borrow a slide from my colleague Pat Levitt. This is showing the change in age is on the x-axis, of normal brain plasticity that's influenced by experience. And this is showing the physiologic effort required to enhance neural connections. And so the sad part of this is that you can see that plasticity is very high early in life and starts to decline dramatically over time. And one reason for that is that the effort involved in changing these connections increases astronomically over time. And sadly, there's a crossover point someplace around here where, as we say, nothing good happens after the age of 30. So let me give you an example, my only perhaps humorous example in my talk, of the advantage of acquiring a skill during a sensitive period. So this is me teaching my 2.5-year-old son to play squash. Now, I played squash my entire adult life, but I did not start to learn when I was 2.5 years old. So in the next frame, I'll start this in just a moment. I want you to, this is now 25 years later, and with my son and I were playing double squash against another father and son pair. My son and I are dressed, have black t-shirts on, and then you can see the other father and son pair. And pay close attention to the fathers versus the sons. We'll start this one first. So that's the other father, and then my son, and that's me getting out of the way. That's my son there, and the other son, and then my son, and then the other son, and then my son. If you see here, my son, and then the other son, and then my son. The point there, of course, is what the fathers are doing versus what the sons are doing. The fathers are providing encouragement and emotional support while the sons are actually doing all the running. And the point is that acquiring things earlier in life and then if they continue often leads to greater mastery. And we often can use sports examples of this. And those of you who play golf have seen the pictures of Tiger Woods swinging a golf club when he was 2 or 3 years of age. So let me close out this part of my talk by talking about the future. Can we reverse critical period plasticity in adulthood? And this is, again, I'm borrowing from my work by my colleague, Takao Hench. And that what this is arguing is that if a critical period is closed in the first few years of life, for example, is there a way to rescue it later on in life so that you could change behavior? And we can think of, in the sphere of psychology, psychotherapy might be one example of that. But in a moment, I'll give you an example from psychopharmacology of another way to do this. So briefly, many elements of postnatal brain development adhere to this principle of critical periods. Most critical periods occur during a time of rapid brain development, generally the first years of life. After a critical period is passed, the effort involved in altering the brain becomes greater. Two big questions are, can we figure out a way to rescue a critical period in adulthood? And are critical periods limited to the first years of life? So something that's being debated over the last few years is whether puberty brings with it another critical period in brain development. Sarah Jane Blakemore at Cambridge has made this argument. I personally think that adolescent brain development is a hallmark of that is elasticity and plasticity, but not a critical period the way we think of ocular dominance columns or speech perception. So what do we know about the biology of critical periods? Well, we know that most of what we know comes from sensory systems. We know that the maturation of inhibitory neurons, like parvalbumin expressing cells, drives critical period trajectories. Importantly, the maturation and the maintenance of these PV cells is sensitive to experience. So these inhibitory neurons are exquisitely sensitive to experience, and without experience, the number and the strength of these PD synapses onto pyramidal cells is reduced. Critical periods most likely close to help stabilize a system that developed during that critical period. And this closure is due to molecular breaks like perineuronal nets, which wrap themselves around the PV cells, and then they restore an excitatory and inhibitory balance. So the point we're making here is that if you ask, why do we have critical periods? It's a way for the brain to become stable. Imagine if we didn't have a critical period for vision or language, and our whole life we were subject to remodeling that brain due to experience. So that's why we think we have a critical period. Finally, the balance of local circuit excitation to inhibition, or what we refer to as EI balance, can, through drugs or sensory experience like deprivation, can shift the timing of critical period windows. And finally, we know that if you lift these molecular breaks, for example, you dissolve the perineuronal net, you can rescue or reopen a critical period. So on that note, let me now turn to examples of how the experience actually influences the developing brain. So we have experience-independent development. Here we simply mean experiences are required for development to proceed in a typical fashion. And this is something that psychologists often refer to as innate. Experience-expectant development. Here I'm drawing from work from the 1970s and 80s by Bill Greeno, particularly. Development is based on the expectation that that appropriate environments will provide information needed to select appropriate subsets of synaptic connections. And this is common to all members of the species. So the argument we're making here is that genetics provides a basic blueprint, but on the assumption that all members of the species will have common experiences, hearing sounds, seeing things, and the like, we leave it to experience to sculpt those synaptic populations. And experience dependent is unique to the individual. This most likely involves the active formation of new synaptic connections throughout the lifespan based on each individual's interaction with his or her environment. And learning and memory is the classic example of this. And I often use the example of my 90-something-year-old mother who uses a computer and a cell phone and an iPad and other forms of technology, although she didn't grow up with this and only learn this late in life. And that's an example of how the brain can still change when you're later in life. And here I would also refer to an excellent paper that Lindenberger and Lugden published not too long ago in the Annual Review of Developmental Psychology. So experience-independent development. The classic example, of course, is Lorenz's ducklings. And so here we have imprinting. No experience is necessary. These ducks were born to follow some conspecific. In the remainder of my lecture, though, I'm going to focus mostly on expectant development, drawing from visual development, auditory, speech, language, face processing, and if we have time, social, emotional development. So we'll start with vision. We know that stereoscopic depth perception or binocular vision, the ability of the two eyes to fuse an image to create the sense of depth, is dependent on activity-dependent elaboration of ocular dominance columns, which is driven by experience. So the actual formation of the columns is experience independent. They just form based on a genetic script, but their elaboration and function is highly experience dependent. And a proper eye alignment does not occur by the end of a sensitive period. Binocular vision will be permanently compromised. And the case study for this has been the study of infants born with cataracts. So Daphne Maurer and Terry Lewis and others have studied this for some years, but Maurer and Lewis have done this for longitudinally for many years. And here's a bit of an older paper, but they report that children with congenital cataracts, at least for low-level vision, the aspects of vision that develop the earliest are the least likely to be adversely affected by abnormal visual input, whereas those that develop later are more severely affected. So for example, when vision is restored, and my colleague at MIT, Pavan Sinha, has done this work in India, when vision is restored, motion perception may come back, but face perception may in fact be altered permanently. Early visual input is necessary to preserve the neural infrastructure for later visual learning, even for visual capabilities that do not appear until later in development. So again, we need that early visual experience. The later the cataracts are removed, the greater the consequences with visual development. And we've known this for years. In fact, if we see over here a very old slide, here's Hubel and Wiesel, who made the initial discovery of these ocular dominance columns and plasticity. We can see here that the older the child is at the age of surgery, the worse their vision. So if the surgery to correct vision is done very early in life, then there's much more likely to be a complete or near complete restoration of binocular vision. But the longer you wait, the less likely that is to be the case. We turn our attention to hearing, here's an interesting case study in auditory development. So this study, and there are others as well, of course, focused on language learning among deaf children receiving a cochlear implant. And they looked at 3.5-year-olds and 4.5-year-olds who had received their implant by their 3rd birthday. And what they reported is that language scores increased with younger age in implant, even when controlling for the duration of implant use. So children who received the implant at the youngest ages obtained language scores comparable to those of hearing age mates by the time they were 4.5 years of age. But children who were implanted after 24 months of age did not catch up to their hearing peers. So the conclusion is that children who received a cochlear implant before substantial delay in spoken language developed between 12 and 16 months were more likely to achieve age-appropriate spoken language. So the earlier the implant, the better the outcome. There's long been this discussion about, well, if you are deprived of in one sense modality like vision or hearing, do you compensate in others? So one way this has been looked at is, do deaf individuals actually see better? Well, they don't see better generally, but we do know that visual orienting, for example, moving your eyes to catch an object in the periphery and reorienting is better. Visual motion processing is better, and visual stimulus onset direction, knowing which direction the stimulus is coming, are all a little bit better in deaf individuals than in hearing individuals. And finally, this is an old paper, but I think it nicely makes the point I'm trying to make here, which is that language abilities of adults who learned English or American Sign Language at different ages have different outcomes. So I'm going to walk you through these two studies, study A and study B, in just a moment. So in study A, there were children who were born deaf, had no experience with language until they learned American Sign Language when they were school age, between 9 and 15 years of age. And so on the y-axis is their task performance. And you can see in this first here, these are the children who were completely auditorily deprived until they were school age. And you can see how poor their language performance is. But in the next bar here, these are children who were born hearing. They heard English from infancy, and they learned sign language after becoming deaf when they were 9 to 15 years of age. And you can see that they have very, very good performance. In the B group, these are children who were born deaf and had no experience with ASL until school age, 4 to 13. And again, you notice that although it's better than here because they were slightly younger, it's still only at about 50%. Whereas these were children here who were born deaf, but they learned ASL in infancy. And these are children in the far bar who were born hearing. They did not hear English in infancy, but they learned English at school age. But again, their performance is quite good because of the auditory input. So the conclusion then is language development is compromised without early language exposure, and the timing of early language influences later language learning capacity. I think the most amazing example of the plasticity of language systems comes from children who've experienced a left hemisphere stroke at birth. And this is very new work by Alyssa Newport. I forgot to put the reference here, but I think this work was published about a year ago. So we know that in healthy adults, roughly 95% of the population is left lateralized for core functions of language. And this is probably also true for left-handed people. A major left hemisphere stroke to the language network in adults will leave you permanently impaired in language, an aphasia of some sort. But what Alyssa has been asking is what happens when the left hemisphere is injured early in development? So here's a classic example of a left hemisphere stroke in this old scan. And you can see the areas of white represent that most of the left hemisphere has been atrophied because of this disruption in the blood supply. These are functional images, and what you're looking at is that perinatal left hemisphere middle cerebral artery stroke, which is a major cause of these strokes. Notice that in all 6 cases, these are 6 different individuals, it's regions in the right hemisphere that are activated when hearing language. So here's a very large stroke in the left hemisphere where these are smaller strokes, but in all cases, language is moved to the right hemisphere. But you'll notice over here, where this is the least damage on the left, there's still a little bit of language in the left hemisphere. And if you look at this behaviorally, we're looking at sentence comprehension in green and word structure in orange or brown. And this is accuracy. So in the far left, there are siblings who are healthy controls. These are left hemisphere perinatal strokes and right hemisphere perinatal strokes. And in all cases, basically what you see is that the children's language performance who had the left or right hemisphere stroke is almost identical to the children who did not have a stroke. So even without most of the left hemisphere, children develop normal language if the injury occurs very early in life and the homologous region in the right hemisphere takes over language function. So now let me turn to the development of speech and language. We don't recognize speech and we don't acquire language without hearing speech or language. But as with vision, the brain likely is pre-wired to benefit from that experience. And with experience comes specialization. So we have known for decades by studies by DeCasper and Pfeiffer that newborn infants can recognize their mother's voice, and they do so based on the experience of hearing their mother's voice in the last weeks of pregnancy. So they recognize and prefer their mother's voice versus a stranger's voice. They distinguish between a mother reading a familiar nursery rhyme, one they heard prenatally, and an unfamiliar one. But they don't recognize the father's voice, in part because they don't, much of the sound is conducted internally. So this is some DTI, diffusion tensor imaging data, showing what the foundations of these networks for language in these two pathways here, what it looks like in the newborn and what it looks like in the adult. And clearly you can see it's much more detailed and elaborate in the adult, but it is present. In the longitudinal fasciculus and the arcuate do exist in the newborn infant. So we know now that the timing and the nature of experience with language impacts the perception of language. And so now I'm going to be heavily drawing from the work by Janet Worker. So we know, and we've known this for decades, that before 9 to 10 months, infants can discriminate the sounds of all languages. But by the end of the first year, they're only able to discriminate the sounds of the language in their own hearing environment. And that's what you can see in this graph here. So this is what we often refer to as perceptual narrowing. You become an expert at recognizing the sounds in your environment. But prior to the critical period closing, you can discriminate the sounds from any environment. So an English being brought up in a German-speaking home at 6 months can also discriminate the sounds from Japanese or Czech or Thai, but by a year, they've lost that. Pat Kuhl has demonstrated that if you give 9-month-olds 5 hours of exposure to a non-native language from a live tutor, then you can recapture this ability. This exposure has to occur via audio or videotape. Sorry, this exposure has to be a live tutor. If it's done by audio or videotape, you don't see the effects. And you see the same thing in songbirds. Songbirds need to learn song from the father, but a live tutor. And if they only hear the father or see a video of the father, song is abnormal. And we know that there are functional impairments here, that the perception in infancy predicts later vocabulary size. So the better you are in infancy in discriminating sounds, the better you are, the bigger your vocabulary as an adult. And this goes back to a point I made earlier about cascading critical periods. And so here, Janet Worker and Takao Hench have basically shown here that there are cascading critical periods. So even within language, there are different critical periods. So language discrimination occurs the critical period occurs early, native phonetic categories, word forms, so on and so forth. We know that much of language acquisition occurs in a face-to-face fashion, that infants use visual information in talking faces and imitate the sounds that they hear. I'm sorry, oops. Well, I apologize for this. I will have to, as many of you know, much of the United States, particularly in New England, is still on lockdown. And as a result, everyone is working from home, and we have all discovered the limits of bandwidth. And so we have been finding that we're running out of bandwidth. So just give me a second while I go back to this. And I certainly hope I'm still, okay, I'm still online. All right, so let me go back.
You are still online, and we can still hear and see you.
I have to reload my talk.
No problem. We are used to having technical difficulties with all these virtual meetings going on in between.
And I'm nearing the end of my talk as well. So there we go. All right, so.
Okay.
Okay, so to summarize, the neural organization is in place by birth for learning language. The perceptual systems provide the first entry point, that babies are open to different aspects of language at different points in development, that critical or sensitive periods for both heard and seen speech exist. And the slide that crashed, and I I now realize I had several movies in that slide, which is probably behind this. What that was showing is that Janet has shown that babies can discriminate speech sounds even when there's no auditory soundtrack, just looking at the face. So in English, they might be able to discriminate ba from ga by reading the lips. And the point here is that speech perception and language acquisition often occurs in the context of face perception. Targeted experiences and exposures can change the timing of critical periods, and changes in timing may have implications for language acquisition, language use, and later literacy. And I think what I'm going to do in the last few minutes, I'm going to close by now briefly talking about face processing, and lead with the question of, are faces special? So there's evidence in the adult that face and object processing are dissociable, both behaviorally and neuropsychologically, that selective brain lesions can impair face processing, that in monkeys there are specialized areas for processing faces and parts of faces. In the human adult, faces also appear to be processed in specialized areas, and across multiple species, there's a preference for face-like stimuli that are observed very early in life. So let me just give you one example from some work we published some years ago now, which is how specific is face processing. So despite the improvements in face processing observed over the first 6 months of life, we still have to ask how specialized and specific this ability is. And one way this has been looked at is by examining the processing of what we call other faces, other race, other species, other gender. There's any number of phenomenons. I'll talk about other species. So what does the other species effect look like? So in a paper we published now many years ago, we asked 6-month-olds, 9-month-olds, or adults in a simple visual preference paradigm to discriminate 2 human faces or 2 monkey faces. And what we find for human faces is that at every age, 6 months, 9 months, and adults, they are very easy, they have no difficulty discriminating a novel human face from a familiar human face. But if you look at the monkey task, only the 6-month-olds could discriminate the 2 monkey faces. That ability was lost by 9 months and not present also in 12 months, and in adults, I mean. So similar to speech processing, there's a perceptual window that narrows with development. So a follow-up study was designed to ask the question, can you keep this sensitive period open with additional experience? So in this study, we did a training study, and infants went home. We pre-tested them at 6 months. Again, the same thing of, can they discriminate 2 monkey or 2 human faces? And then one group went home with a picture book of monkey faces, and one group did not. And then they were retested at 9 months. And so every day in the intervention group for 3 months, parents every day spend time going over and naming these monkey faces. And what you see is that in the 6-month-olds, we see the same robust effect, that there's a preference for the novel face. So before intervention, all the infants could discriminate 2 monkey faces. So we replicated the earlier findings. The 9-month controls who did not get the intervention cannot discriminate the 2 faces, same as we've reported before. However, if the infants in the intervention got the training, they could discriminate the faces, and moreover, they could generalize it to monkey faces they had never seen before either. And so here are here's the slides there. What was not reported in our original study was that we did a training study in adults, and what we showed is that even adults who worked with monkeys on a full-time basis, although they could discriminate the monkey faces in their colony, when you tested them with monkey faces they had never seen before, they were no better than chance. So the other species perceptual window closes at around 9 months unless additional experience is provided. But this experience must occur during a sensitive period, and even adults who have extensive experience with monkey faces are not as good as infants if that experience was acquired as an adult. Let me close on a monkey study to confirm this. In an original study by Sugita, they reared monkeys from birth without access to faces. The deprivation lasted from 6 to 24 months. When they tested them immediately after the period of deprivation ended, all the monkeys, regardless of deprivation, could discriminate 2 monkey faces or 2 human faces. But then, at that point, one group saw only human and one group saw only monkey faces. So they gave the monkeys experience with either monkey or human faces. And from that point on, they could only discriminate the faces from the exposed category. So all deprived monkeys could, at the minute the deprivation ended, could discriminate to monkey or to human. But then the monkeys that were then exposed only to monkey faces soon thereafter lost the ability to discriminate to human faces, and the reverse for monkey faces. In a more recent work by Marge Livingston here at Harvard, she studied two groups of monkeys, one reared from birth seeing faces and one reared from birth with no face exposure at all. And then after they were 90 days of age, she presented them with images of faces, hands, and objects while performing functional MRI. And the results was normal retinotopic organization for objects and hands, but not for faces in the face-deprived monkeys. So similar to Sugita, exposure to faces is a requirement for face expertise to develop. So collectively, it appears that the infant brain possesses a general neural substrate for all-purpose face processing, but this face specialization is entirely, we think, experience dependent. The perceptual window through which faces are viewed is broadly tuned early in life and narrows with experience. The unknowns, what's the nature of the experience? What do infants actually have to see to drive the system? When does the experience have to occur, and how much experience do they have to have? So let me close with just what we don't know about critical periods in human brain plasticity. Unlike animal models, we really don't know much about the dose, timing, and duration. That is, what specific experiences at what dose and at what time periods are needed to drive development. Most of what we know from this is from studies of experiments of nature, children with cataracts or born deaf, but there's still much we don't know. We don't know how broadly or narrowly these critical periods are shaped. There are very few studies that have attempted to manipulate critical periods. So what is the ideal, which is really the ideal way to understand biology? So intervention studies, such as the one my colleagues and I have done on the Bucharest Early Intervention Project, where children experienced different degrees of psychosocial deprivation, but then one group was placed into an intervention. This is also a way to understand mechanism. And then there's the question of what accounts for so-called sleeper effects. So you don't see the effects of early experience until many years later. And this is a very old phenomenon, and we still don't fully understand that. And finally, I think an area of active investigation now is how do we rescue critical periods in adulthood? And Hench and others have shown that pharmacological interventions can in fact rescue critical periods. Unfortunately, these interventions are usually GABAergic drugs, such as the anti-epileptic drugs. And then there's always the issue of not only safety, but you're not opening a critical period for one aspect of language, you're opening up critical periods in the brain generally. So there's clearly a downside to this. So on that note, I'll thank you for your attention and close out my talk and then ask if there are any questions. So thank you.
Thank you very much for these very dense and rich presentations. We are very much looking forward to the discussion now, and as it is a common tradition in life, we would like to open the stage first for our fellows and to give them the chance to ask the first questions. So in this case, I would like to ask one of our panelists, Christy Ching, to start. I can't hear her at this moment. Also here, it's sometimes difficult with the technical parts. Okay. Yeah, now it works, yeah.
Sorry about that. Thank you so much for the lecture. I had a few questions. I thought it was really interesting when you talked about exposure having to be live, like for like you can't use audio. And I was wondering now that maybe it has to do with like having the facial perceptions, but maybe now with like the people are using technology so much, like maybe they put an iPad in front of like an infant, or they distract the infant with TV, if that would be maybe detrimental to like their learning, like if they're not using enough in-person or face-to-face interactions for infancy. Do Do you have any
So,
thoughts?
you know, it's an excellent question. It has not been exhaustively studied, but the little that has been studied confirms your expectation. So we know that songbirds who learn song from a video or an audio tape have abnormal song. And there is evidence that children who are exposed to large quantities of television and not human interaction also develop atypical language. So if I dig a little deeper in your question, then there's the issue of, so what is it about the social interaction that is such a potent driver of development? Some of it is that we're social beings. Some of it may be the multimodal information, which I think was also something you said. So you see the face and hear the soundtrack at the same time. But clearly more work needs to be done to flesh this out a little bit. You said you had 2 questions, I think.
Yes. So I was wondering about, would there be any disadvantages to keeping the critical period open or the perceptual window? Because I think, like, I was just wondering, like, what are the advantages and disadvantages of trying to extend it or move it?
I think the argument that's been made is that if you extend the critical period, the system can become unstable. So rather than forming a system that allows you to discriminate speech sounds, it's constantly being refined and remodeled, and that can lead to instability. But of course, that's a thought experiment because no one's ever done this. Takao and I have a collaboration where we're looking at babies who are exposed to very high levels of GABAergic drugs for very long periods of time. So these are complex surgery cases where the children are heavily sedated with GABAergic drugs and anxiolytics for sometimes weeks at a time. And the question is, does bombarding the brain at 3 months or 6 months with these drugs shift critical periods? And I don't know the answer yet, but we will in a few months.
Thank you.
Thank you.
Thank you very much. The next is another fellow of life. It's Andrea Hasel, please.
Okay. You see me?
No.
No. Okay, well, but you hear me, right?
Yes, yes.
We hear you, yes?
Okay, great. Thank you very much for your talk. It was very, very interesting. So I was wondering, you were talking about this critical phase when babies are 3 months old and they can discriminate between monkey faces, between different monkey faces, right? But they lose this ability later on.
Right.
And I was wondering, because there are those companies out there who make money with claiming, okay, you have to promote your child's abilities as early as possible. So I don't know, get them out there with 3 months and teach them a foreign language and so on. So I was wondering, what do you think about these offers?
Yeah. So the classic example of that was a company called Baby Einstein. And this was a company that was doing exactly what you said and arguing that, if you plop your baby in front of a tablet or a television and they heard French and German and Spanish and English spoken and they saw colorful shapes, it would make them smarter. There was not only no evidence for that, but eventually the company was sued for making false promises. So the reality, going back to the first question, is that infants need social interaction. And they don't get that from these other media. Now, an interesting sub-question would be using FaceTime or Zoom, where there is an interaction, but that's different than companies that were simply showing video material. This kind of interaction, we're now going to have a whole generation of babies who spend an enormous amount of time doing this kind of interaction, and will that have any impact on them or not? But of course, keep in mind that they are also exposed to conventional social interaction. This is just an addition. Imagine a baby who is brought up only on FaceTime, what would happen? Yeah. So it's the nature of the didactic interaction that we think is important.
Okay. Okay. Thank you very much. That's very interesting. Thank you.
Thank you very much for this question. We continue with our fellows, and on my list now it's Sarah Polk from the MPI.
Yes. Hi, can you hear me?
Yes.
Yes.
Great. Yeah, thank you. The talk was super interesting. I had a question about the cascading critical periods. You briefly mentioned this when you talked about the babies who were exposed to a lot of different sounds during their critical period, that the babies who were better at differentiating between different sounds in any different languages were then later, they showed higher vocabulary skills, essentially. Are there follow-up studies on adults who have maybe high phenome exposure when they're very young, or maybe going back to your squash example, are there follow-up studies on adults who as children had a lot of training, maybe they had a lot of sport training, they had a lot of experience learning how to play sports or learning different languages? Are there like later life transfer effects, quote unquote? I don't know if you can call it that.
Yeah, great question. I'll address it by citing one paper and then one discussion. So we think of this as reactivation. So the example would be if you're exposed to something when you're a year or two, or even for the first 2 years, but then no exposure until you're an adult. So there is some evidence to suggest that if you were exposed to a second or third language early in life for some period of time, like the first 2 years, but then you're not exposed to it again until you're an adult, you can acquire it more quickly if you have that early exposure. So the way we think of this is that you built the network for, say, a second language. And even though many years might go by without using that network, the network still exists in some form and can be reactivated. Mm-hmm. And so what we don't know is, does that mean, thinking of a family in Germany who is exposed to German and English, an infant exposed to German and English for the first 6 months, but they don't hear English again for 20 years. Will they be able to acquire English more rapidly, or did they really need to hear English for the first 2 years? And we simply don't know. But the assumption is that those networks that are built early, if they were pretty well established, don't completely disappear, and they're capable of being reactivated later on. But this is, again, where an animal model would really be the way to go. I hope that answers your question, because it's a great functional question of, if you have these early experiences, can you reactivate these things later in life?
Yeah, I think my question, I mean, this also answers part of it, but I think my question was also kind of going into the realm of transfer. So if maybe a child or an infant was exposed to English and German, and they only spoke German growing up, but they had 2 years of English experience while they were really, really young, would they then be able to acquire a different language then more quickly, or would this not help at all?
Yeah, I don't know if we know that. I mean, that's a great question. So you did say the word transfer, and I apologize for not catching that. So would they, for example, learning another Romance language like Italian or Spanish or something like that, if they had already mastered 1 or 2 Romance languages? I don't think we know. Talking to people in the language area, they would argue that if you have built a foundation, here's the best way to put this. If you have mastered a few languages early in life and they've been maintained, the critical period may not close for language acquisition, and it may be much easier to acquire a third or a fourth or a fifth language later on. But again, this is a discussion we've had. I can't point to any papers that support that.
Yeah, no, it's a really interesting thought. Thank you.
Thank you.
Thank you very much. Now I have the last question, our fellow first round from Mareike Trauernicht. Afterwards, I would open the floor also for the rest of the audience. So now please, Mareike.
Yeah, thanks for your talk. I know that you had to choose some domains, but I was wondering about the domain of cognitive development. And if you could say a few more words about also critical periods in cognitive development, and how we might be able to rescue them also in later life, especially in later adult life, like when older people are really suffering from cognitive decline.
Wonderful question. I actually purposely avoided a discussion of cognitive development because it's so difficult. So here is a way to back into that. We know from studies of early intervention, like in the United States, Head Start or Abecedarian, or the famous study done in Michigan, and I said it that way because I immediately forgot the name of the famous study done in Michigan. We know that children who at least grow up in disadvantaged homes, if they receive early intervention services, which includes cognitive stimulation, they do show initial gains. But we also know that if that's not maintained, so they go into early intervention services from 1 to 3 years, but then after that, they don't get it. They show big gains in those first few years, but when you see them again in school age, they don't show the benefit anymore. So that argues for the maintenance part. The second part is, when we say cognitive skills, what do we mean? This is why I sort of avoided this. So I will draw from an example from this long-running study, this Bucharest Early Intervention Project. So in brief, a large number of children had been abandoned at birth and run in state-run institutions. After a baseline assessment, half got put into high-quality foster care, half did not. And what we showed is that IQ was dramatically increased by the children removed from the institution and put into foster care. But the biggest increase was seen by those placed before the age of 2, hence the critical period. But we did not see a big improvement in executive functions, which is something we looked at much later in childhood. But that begs the question of, is that because the circuitry that underpins executive functions, notably the prefrontal cortex, is relatively immature in the first few years? So it would really be difficult to do an executive function intervention in those first few years. And then I get into a conundrum of what that would look like. So, for example, inhibitory control. How would you, 1-year-olds and 2-year-olds just don't have good inhibitory control. So what would an intervention look like? So we think there are some critical periods in cognition, but we just don't know that much about them. And then the second part is, would there be some advantage of early cognitive stimulation that confers an advantage much later in life? And so there are data from the Alzheimer's literature showing that the prevalence of Alzheimer's is lower among those who finished high school or more than high school, with the argument being that the cognitive stimulation that occurs confers some advantage or protection. But what we don't know, is it because of 20 or 30 years of education, or is it because they had that education in the first few years? And we just don't know that. So I hope that that's the best I can do in answering your question.
Thank you very much. I would also have a long list of questions, but the list of people who are interested in your comments is very long and increasing still. So I just put myself in the back, and I would like to ask Meltem Yüksel to go on for the next question.
Hi, can you hear me?
Yeah.
Thank you so much for this great talk. So my question is about the current pandemic situation, because most people have been isolating themselves, especially if they're new parents. So even if these parents of newborn kids during this pandemic have been getting some sort of language input from their parents, they're not really exposed to the normal variety of language around them. How do you think this might affect what we would normally think as like a typically developing kids?
Let me back into that with an example, again from this work in Romania. I predicted, the very first hypothesis I had was that face processing would be fundamentally altered in kids experiencing psychosocial deprivation, because they would have very limited exposure to interacting with faces. And it turns out I was absolutely wrong. Every way we've looked at face or emotion processing seems to be preserved in these kids. So the argument we've made is that they had enough stimulation in the beginning to get this system up and running. So in terms of your question, my prediction would be they may not need a range of hearing many different speakers talking, as long as they have normal language input, even if it's from only one speaker, it will generalize, which goes back to the earlier question about generalization. So if an infant from birth to 9 months only hears mom or dad speaking, my intuition is they'll have perfectly normal language, that they don't need a variety. But sadly, this is an experiment of nature, and we'll actually probably know this in a few months. Hopefully, we'll know in a few months. We don't want to be in our houses for another 2 years.
Thank you.
Okay, thank you very much. Now we continue with Alexandra Freund, one of our faculty members in the live research school. Welcome, Alexandra.
Thank you. And thank you also from my side. It was a really fascinating talk. I, of course, as a lifespan psychologist, sort of perked up when you said it's all gone after the age of 30. And I would like you to constrain this maybe a little bit and refer to your squash skills and not have this the main message of a talk. Which of course you didn't. This was an anecdote, of course. But I would like, on a more serious note, ask you about sensitive periods as they might apply to adulthood. Is there anything like that in adulthood? And I mean, broadly speaking, beyond the age of, say, adolescence, in the sense of a window during which a certain skill or ability can be acquired. And
Right.
Would it make sense to you also to think of a sensitive or critical period as a window that ends at a certain time? So is there an age after, until which you have to have done something in adulthood, and if you don't afterwards, it will decline? So not that you don't acquire something, but you can't maintain it.
All right, let me unpack that. There's a lot in that question. So let me first walk back my slide of the dramatic drop in plasticity. When I talked about experience, expected, and dependent, I made the point that experience-dependent development, the classic example of that is learning and memory. So there seem to be no critical periods regulating that, although I will come back to Marika's question about cognitive stimulation in a moment. So what that's saying is that if you think of this from an adult learning perspective, there's no reason, assuming there's no underlying pathology, that learning and memory isn't something that will continue throughout the lifespan. It may be that there are foundational skills, of course, that if you don't have them early, it'll be hard to acquire them later. So the classic example is the differences in kids with good executive functions in childhood do better in adulthood, because you can, if you have poor executive functions and you're 4 or 5 or 6, it may be difficult to acquire them when you're 30 or 40, like inhibitory control and planning and cognitive flexibility. But there I'm speculating. I can think of an example of the last part of your question about, we know about maintenance, but what we don't know is, are there simply, well, there probably are things that are difficult to learn as an adult compared to when you're younger. So I briefly decided I wanted to learn, when I first started doing neuroimaging, I thought I would learn or relearn physics. That didn't happen. I found the physics very hard. As I started thinking about genetics, I thought I could learn biochemistry and molecular biology, which I didn't have much background in, and it turns out I really can't. So that's what I mean by foundational skills. You know, when you learn chemistry or math, you need to start at a simpler and move to a more advanced level. So I think the argument is, if you didn't get very far, if you only got as far as algebra, doing number theory as an adult might be difficult. But you know, there's probably a literature here that I'm not familiar with. One thing I could offer, I think, is Nadine Gaub is a colleague of mine, and she and I teach a class, and she gives one lecture on training and adult plasticity. The question being, are there companies out there, for example, making technology that will allow you to become smarter as you get older, or to make sure you don't lose your memory capacity? And the findings are very sobering. There seems to be virtually nothing out there. We can talk about the virtues of exercise, but when it comes to things, you know, companies that are making brain training games, the evidence is rather weak that they actually are efficacious and generalize. So in the end, I'm not sure I really answered all of your questions because I took them apart and then I realized I may have gotten lost in each one of the pieces. So come back at me if I didn't answer your question.
Thank you very much. We are now calling for the last, very last questions. So if you have one last burning questions, please let the organizers know. For the next question, I would like to ask Robert Scholz.
Okay, hi there. So I was wondering what you would think the role of neuromodulatory systems such as serotonin, dopamine, and stuff like that is in gating this plasticity. So, I mean, you presented results on the GABAergic circuits and things like that. So, yeah, what's your take on that?
Yeah, great question. So my expertise is a little thin, but as I talked about inhibitory neurons, I realized, you can shift excitatory inhibitory balance with excitatory functions as well. So one way to approach your question would be, although this GABAergic system may have an impact on closing a critical period, really what is at play here is the balance between excitation and inhibition. So you can shift that balance by also acting on the excitatory system, not just the inhibitory system. Did that come close to answering your question?
Yeah, somewhat, of course, yeah.
Thank you very much. And now we come to our last question for the session today. And here I would like to ask Ulmer Lindenberger to pose this question.
Can you hear me?
Yes.
Yes.
Yeah. Great
I hope
talk.
this is an easy question.
So I completely agree with this lifespan view of plasticity that you showed as a cascade of critical periods where one builds where the later ones build on the earlier ones. I think that this is a very compelling picture. And also, I think that these differences between experience independent, expectant, and dependent forms of plasticity make sense. Now, in adulthood, we do have more, what you may call more local plasticity, like in the course of skill acquisition. And we know that these forms of learning are also accompanied by structural changes in the brain. And I think the question I have is whether, despite the differences in amount and of course also the difference in function, because it makes a difference whether you build a system or whether you acquire a skill on top of an already a mature system, whether despite all of these differences, it may make sense if you go to the mechanistic level to search for general principles such as EI balance, GABA inhibitory neurons, perineural nets, and so on, that may actually govern both these more local forms and the ones that really shape development. And I'm just curious what your intuition is about that.
Yeah, so excellent question. You and I can continue this through the CFAR network as well, but I think you're right. And I think there's probably a common set of biological mechanisms that regulate this across the lifespan. And that does bring to mind the last question that was asked, because there was one part I forgot to mention, which is, are there examples of critical periods later in the lifespan? So when I've had this discussion with people who study adolescence, one thing that I've been worried about is that a strong need in adolescence is social contact with others. And so a natural experiment is what would happen to social communication and social development if adolescents were deprived of social contact. And sadly, that's another experiment of nature. In the United States, it's children who are incarcerated in youth and then placed in solitary confinement. So there are large numbers of children who are 14, 15, or 16 who are in complete social quarantine. And what we don't know is what effect that will have. And I think to put both questions together, The former question would be, are there other examples later in the lifespan where certain experiences are necessary? And the Norman's question really is, are the biological drivers for that any different later on in life than earlier in life? At least that's how I heard your question, Norman. And I think the answer to that is probably no. I think they're the same.
Thank you.
Thank you.
Thank you very much. For the sake of time, I would like to close our session today. I can imagine that there are still numerous questions that would arise from this inspiring presentation and this lively discussion, and we could continue for another hour, but I would like to leave it up to this point, and would like to encourage you to attend next week again when we have Candice Odgers for the next presentation in our lecture series. And again, I would like to thank Charles Nelson for volunteering for this talk here and delivering such a great presentation. Thank you very much.
My pleasure. Thank you. It was a great honor, and I thoroughly enjoyed the questions.